purpose of this video is to give you some clues about how to handle the acid bases and buffers lab. And the first thing you'll be doing is you'll be finding the pH of some different solutions, and what that requires is that you'll get some containers here. I'm just going to do one test tube, but you'll have 6 of them here all along the bench. And what you'll do is you'll put 10 mils in the first one of water, and you can mouse over each of these and see what's in them. And there's water in this one, so I'll take the water down here, put it over the test tube and add 10 mils. You'll do the same for the other things that are required here, 10 mils for each of them. Then it asks you to stick a pH meter in there, and the pH meters are up here, so take that and put it over the test tube. And what you can do is you can zoom in here and you can maybe read the pH meter a little bit easier. The other thing you'll need to do is add some bromophenol blue to each of these test tubes once you've put solutions in them. So you can do this one here. I think it says 2 drops. So mouse, put it over the test tube and then hit two drops here, and you'll see it may change color. In this case, it changes color to this green color. And if you have any issues, sort of seeing the color, you can just zoom in and there it is. So that's the first part. Next thing we need to do is clear the bench, and we'll do this again. Zoom out here. There we go. And what we're going to do is check out some phosphate buffers and what we need to do with those. So we get these 4 test tubes from the shelf and we put these different things in them. And again, it's just going to be the same kind of story. So we've got our test tube 1 there, we'll put water in that, and then we'll put sodium monobasic phosphate in the next one, sodium dibasic phosphate in the other one, and then a combination of those two in the last one. You've got to be a little bit careful with the phosphates here. Make sure you're getting the right one. So this first one's water, so you just need 5 mils of that. Then we need 5 mils of sodium monobasic. That one is over here. You can read it says sodium monobasic phosphate. That goes there, so that needs 5 mils as well. The sodium dibasic's over here, and we'll get 5 mils of that as well. And then in this one we do 2.5 mils of each of those. Whoops, 2.5. There we go. You can see each time it fills it up a certain amount. For 2.5 it fills up half what it did for the 5, and there you go, now it's all full. So now we've got all those things in them, and what we're going to do is we're going to place a pH meter into each of these, and we do that from the instrument shelf. Let me move that over a bit. It's probably a good idea is to put these fairly far apart so that they don't get interfered with. There we go, it's better. And again, you'll be able to zoom in and look at those pH meter readings on each of them. Now, what we're going to do here is we're going to look at the response of each of these to hydrochloric acid. So what we need to do is get a clean beaker from the containers here. So we get a beaker, and then we put in 10 mils of 0.5 molar hydrochloric acid, and there we go, 0.5 molar hydrochloric acid, so that goes in here, and we're going to put in 10 mils, and then we get the dropper, and that's in the containers section, and we stick it into the HCl, and what should happen is you get some liquid drawn up into the dropper. Now we can grab the dropper and put it over the test tube, and then we can drop drops in. Now what I'm going to do is I'm just going to drop what it says here. I think it says 2 drops. And hit OK. And then we should observe possibly a change in pH, and we do here as well. You'll do that for each of these. Obviously you'll be putting these results into your lab notes, and then you'll be able to write them up into the experiment. So make sure you put down first of all what the pH was initially, and then what it was after you added the 2 mils of hydrochloric acid. After that, you're going to repeat the procedure with sodium hydroxide, but what you'll have to do is you'll have to wipe everything off the bench before you do that, and then restart it using the NaOH. Next we're going to be looking at the buffering capacity of the phosphate buffer. So we'll do what they ask us to do here. We're going to make up that buffer solution, 2.5 mils of the dibasic and 2.5 mils of the monobasic. Just like we did before, drag it over, add the prescribed amount. There we go. Now it's asking us to take a pH meter from the instrument shelf. We'll stick that in. There we go. And then we do the same thing with the hydrochloric acid. That we did before. And this time we're going to put 20 mils of the hydrochloric acid in the beaker. That's what it asks us to do. And then we'll get our dropper, and we'll stick it into the acid. Of course it'll fill up. Now you'll notice that we've started out here with a 7.21 pH, and if you don't see that, that's fine. You can zoom in when you do it, because that's what's going to be important. It's what it looks like when you do it. And I've already done this, and you can see that before you add any HCl, 0 drops initial, the pH is 7.21. Now what we're going to do is we're going to investigate what the pH is after doing drop by drop of HCl. Now then we're going to do the same thing with the sodium hydroxide and do drop by drop for that as well. Of course the 0 initial is going to be the same for the sodium hydroxide as well, but these of course are going to be different. So here's how it works out. And we'll stick the thing over the dropper over here, and I'm just going to put in 1 drop, and that's what you need to do as well. Just put in 1 drop and hit OK, and then you'll see that the pH changes. So it was 7.21, now it's 7.12, and then we repeat the process, just adding another drop. It drops to 7.03, then 1 more drop and it drops to 6.94. Then what we do is we clear off the shelf here, and we do the same thing for sodium hydroxide. We have to clear the table, grab a test tube, and then we do the same mix as we had before, 2.5 mils of each. Dibasic, monobasic. We do the same thing, we get our pH meter. There you go. Containers, beaker, material, sodium hydroxide, there it is. Again, 20 mils of that. And a dropper. And again, we do the same thing, but we just add 1 drop at a time. So I stick it over, I do 1 drop, and I hit OK, and you'll see again that the pH went up here. It went up to 7.29. We do the same thing again, we add another drop, look up to 7.38, and then 7.47. Now I'll show you in the next video what you need to do with this information in order to make your graph for the lab report.